So this next session is on the biology of the beetles. And I work for the Forest Service, and a lot of the issues that we deal with nowadays are invasive insects and pathogens. And one thing that we've seen with all of these new invasives is that we know very, very little about them when they arrive in this country. The Asian longhorn beetle, the emerald ash borer, we knew nothing about these species when they arrived here. Even species like the beetle that transmits thousand cankers disease, we knew very little about that. And certainly the red bay ambrosia beetle was, nothing more than a few pinned specimens in a collection somewhere. Nothing about its biology. I think we have a little bit of an advantage here with this situation, whether it's Uwelacea fornicatus or something else. We have some idea of the biology of this system with the fungus, and we know what it's carrying. We know something about this whole system. But we still have a lot more to learn. As Yuri said, there's things we know, things we don't know. So what we're going to hear now are a series of presentations on the biology of the insect, of the beetle, and we have a little change in schedule here. Sampath's presentation, I guess, isn't loading properly, so we're going to skip him. And if we have a chance to jump back to him later on, we'll do that. Otherwise, we're going to start with Kiti from Sri Lanka talking about non-chemical and management and chemical ecology of the beetle.
Thank you. Good morning to everybody. Yeah. Now, let me make my presentation basically on some of the non-chemical aspects with respect to the tea shuttle borer, what we have experiences in Sri Lanka. First of all, welcome to the Tea Research Institute of Sri Lanka, a wonderful working environment for shuttle borer, being this particular pest has been in existence for the last several decades. And during this brainstorming session, I'll just take you through two aspects. One is the invasiveness of the pest, and secondly, some of the experiences what we have on non-chemical control. Now, ladies and gentlemen, when you talk of invasive animals for tea, we don't consider shuttle borer, as you consider today, as an invasive pest, because we have been having this problem for ages, and we are on the process of making several attempts. But on top of it, we have some other invasive animals in the tea sector, starting from various other animals in the tea sector. Why I am so concerned about harping on the non-chemical methods in tea, or making the tea shuttle borer control. Tea is considered as the main economic crop for the country, and more than 95% of the tea is being exported, and we have restricted the usage of pesticides while harnessing more of non-chemical methods. So that is the main reason why we are so concerned about the pesticide usage, and tea shuttle borer is one such pest affecting tea, while we have many other pests from top of the foliage down to the root. Okay, now this looks like a a real tea plantation. You can see. It's a fully covered land with tea, and when you go closer, you will see a nice looking foliage full of branches. Per bush, you may get about 60 to 80 developed branches, and per acre, you will have about 5,000 plants, right? 5,000 bushels per plant, right? I mean, for you to think how difficult if this problem is going to be cured if you are harping on one single bush in terms of that way. Right, now on a bush you get different sizes of branches, and you will see young as well as mature branches. And shortle borer, this particular species, will prefer branches of this size, right? Branches of this size, where we say for our convenience we say pencil thickness size branch. So even for assessment of damage, forecasting the damage, we consider these sizes of the branches for our assessment purposes. The damage goes on from young stage up to the mature stages, but not on dry wood, right? Not on dry wood, mainly on the living plant. You can see the damage extended in different life stages of the stem. The first attempt what we do is, as I said, the borer population is found in the environment or in the ecosystem. All what we are targeting is to look for cultivars, resistant tolerant cultivars as the first step, and secondly prophylactic stem protections where we can stop the beetle going into the stem, right? I mean, that is the first step. And then secondly, when we realize the bush infestation, certainly we do surgery pruning, or take off all the damaged portions, and that is another aspect what we advocate. And thirdly, making the plant healthy and improve the plant's vigor through various good agriculture practices. I recall some of the discussions yesterday, expecting the beetle would do a fairly big damage when the plant is under stress, right? So this is one such approach what we practice. So we advocate the growers to go for the good agriculture practices, starting from nursery going up to the mature stage. I mean, cover all aspects like fertilization, taking the harvest properly, and control of other insects and pests or other pest damages below threshold levels. The sanitation pruning I was talking, the tea once in 3 to 4 years time, you take off the branches or mature branches out, and then our main attempt is to make the plant form the plant to a bush, right? So that way we'll take off the branches out, and then, as I said, a surgical operation is done to take off all the affected portions. And in the meantime, take off all the branches what we have taken off, or prune branches, and then chip them and bury them. We expect two objectives. We have two objectives here. One is to take off the inoculum, right? The pest inoculum 0, and secondly, when you incorporate all the chips into the soil, we expect the organic matter level will go up, right? So that's another way of making the plant healthy and vigor. And of course, following pruning or taking off all the affected portions, we do a wound dressing, a simple non-chemical wound dressing. That's a spray lime, right? Just a hydrated lime spray onto the bush, right? And we consider this as, again, when you spray lime, we are experiencing a global change. Climate change now, and this acts as another climate change mitigation, where you don't get splits of the branches, right, or cuts branches that's another objective what we have, and of course we are developing certain other stickers onto this, so that that's going to be a two-way action, while reducing the secondary infections onto the cut surfaces, as well as mitigating the heat stress onto the plant. And I mean, after about say few months time, say 8 to 10 months time, you will get the branches, booming up, and that would be the stage where this, the borer populations would attract to the plant. So that is where we make the spraying, right, just to protect the stem from the beetle attack. Right, now little bit of biochemical and physiological basis in identifying the susceptibility or tolerance of the tea cultivars. You know why you are drinking tea, mainly because of some of the polyphenol compounds, right? I mean, that is what you get. I mean, that's what you expect from tea. So in the leaf you get the same chemicals. So it's the case with stem and the other parts of the plant. The basis, main biochemical basis behind the susceptibility or tolerance in the against the pest in tea is related to saponins, and to a great extent, and also for polyphenol contents in the plant. And there are studies to say that relative amounts of spinasterol and saponin have been related to the pest tolerance, right? And at the same time, this has been identified, or this has been proven to say that some of the substances are inferred with the hormonal activity of the beetle during pupation, right? So I mean, that's something what we know. And we have different, we got about, I would say, more than 30 different varieties, cultivars, with high degree of tolerance, and certainly those plants or those varieties will pose in high levels of these chemicals, right? So that's something what we know, and we are in developing a new variety. We are looking for such biochemical and physiological basis. I'll come to such chemicals later. And some of the another important area where we need to understand is, in between the nodes, right? In between the nodes, normally beetle attack is more prominent towards the leaf, right? Towards the leaf, where you get more of such chemicals, right? More of such chemicals when the borer recognizes the area, so that they come and attack the susceptible portion, right? But in between the internode, you get less of those chemicals, and then you get less damage there. So that's something we know of. And in addition to saponins and spinasterol, we get some understanding on caffeine. Caffeine is low, of course, in tea, right? I mean, mainly you get polyphenols, but polyphenolic compounds, phenolic compounds. But when you have caffeine, then there again there's a degree of susceptibility. And of course, some of the most important volatile compound, right? I mean, tea again, as I said, tea is tea is marketed, or tea is recognized for flavor, right? For flavor, for flavonol compounds, certain volatile compounds are responsible, and there are 5 important compounds identified, and these have been recognized as attractants for the beetle, right? So indirectly, if you are going for a high quality tea variety, certainly the borer damage would be very much high. So it's some kind of a pricing for us to decide whether we are going for a high quality variety or as a balance, fairly high quality and fairly a good cultivar with respect to a high degree of the tolerance. I was talking about this, and infectious stems show increased levels of caffeine again as a defense mechanism, right? In the stem, the levels of caffeine will go up as a defense mechanism. I think we saw some of the examples yesterday here on avocado. Maybe some other chemicals would go up. Maybe in tea only caffeine and polyphenol we have seen after infection the levels will go up. Ladies and gentlemen, now let me give you some success stories with under organic tea cultivation what we have experienced. You know, organic tea has been introduced by Sri Lanka. I mean, Sri Lanka pioneered in producing organic tea, and we have long experience on maintaining pests and diseases below threshold levels using non-chemical methods, of course, without any synthetic inputs. So we have a long-term trial, TRI, Tea Research Institute, organic and conventional comparative trial, where we have observed with respect to shuttle bowl I'm talking now. Under organic systems, we have studied the soluble sugar levels in the stem, and we found that the sugar levels are comparatively low compared to the conventional treatment, right? Conventional treatment where you advocate synthetic methods, right? Synthetic methods or synthetic inputs, fertilizer as well as pesticides. The reason what we noted, the physiological basis what we noted behind this lower sugar level is the stress, right? It's a stress due to imbalanced fertilization in the plant, right? So more water is found or translocated in the conventional system, while under organic systems the water penetration or water translocation is very much low compared to the conventional system, so that the compounds in the stem will be fairly low. And we did a study with the sugar levels and the same extracts, and we introduced the ambrosia fungus, and we noted that under organic system the ambrosia fungal growth is inhibited or low compared to the conventional system. So as a result, even the limited number of beetle would do a fairly big damage under conventional system, while the organic system would do a little damage. And with respect to the important volatile chemicals I was referring a little while ago, you can see in the conventional systems, you can see the level, right? The level or the peak area, I'm just giving the peak area or the levels of these such chemicals, fairly high compared to all these three organic treatments, right? So simply under conventional system, the attraction to borer is high compared to the organic system, right? Organic system where the volatile chemical concentration is fairly comparatively low. So is the case when we did a bioassay with the stems taken from organic as well as from the conventional system, the beetle attraction was fairly comparatively low compared to the conventional system, right? Now the beetle were so keen to go for conventional stems compared to the organic stems. And ultimately, I mean, what is important is how does the damage occur on both organic and conventional stems. You can see, I mean, before I get into the data, we did again, we pumped a dye on the stem, and we noted in the conventional system, the dye went, there were several blocks, sorry, the other way around, this is the conventional, this is the organic. In the conventional system, because of the borer damage, the dye did not move fast, and in the organic system, the dye went without any hindrance, right? The damage levels are very much low under conventional system, and you can see the total damage when you compare with the conventional system. The resultant damage under organic tea and under organic system was fairly low, right? So I mean that was the evidence we have in organic system. And in the meantime, we studied the histological studies or the stem structure in stems. Of course, I mean we studied. I mean we have done this for leaf pest and root pest as well. But this is with respect to a shuttle borer. We noted under organic systems the tissues, the xylem tissue thickness is fairly high compared to the conventional system. That will act another hindrance in making the borer damage lower, right? So I mean that's one example what we have seen. We have seen the same, similar scenarios on leafy pesticide, leaf diseases as well as the root diseases, right? So that's about the histology. To brief you what we have shown or what we have experienced with respect to non-chemical method. Certainly, our main approach in tea shuttle borer management is to look at the critical points where the borer would get into the plant, and then we encourage the good agriculture practices so that the borer damage would be minimized, right? The borer invasion would be minimized rather than waiting for the infected bush and infected plant, so that for us to go for control method. In the meantime, with your understanding here in US, we studied or we went around some of the close by areas where again we saw the pest damage occurring on avocado in Sri Lanka at present, unfortunately, and so is the case on durian, right, and rambutan, right. So I mean those are some of the. I mean of course we knew these plants as hosts, right. I mean we have about 140 odd different plant species as hosts, right, in addition to tea, but we don't consider. I mean we have not seen the beetle completing the life cycle or doing a big damage, but now we have added the host range with these three important plants or important trees to Sri Lanka. And with my presentation, with my experience, right, what I presented now, I think we would help you in studying some of the host range clearance with I mean we were trying to clear the beetle as well as the fungus anyway, but at the same time we should look at some of the host range clearance. Certainly that would give us, the whole team, a good exposure. And of course resistance and tolerance mechanisms in your plants, in your species what you are so concerned about, and also to recognize some of the critical points in avocado cultivations here, right. I mean as we have experience in Sri Lanka, what critical points what you can recognize so that we can advocate or we can target some of the control practices. And of course good agriculture practices for better control, in addition to pesticides or pesticide injections or whatever. And I could not tell you some experience what we have with high potash, right? Using high increased potassium in fertilization or reduced levels of nitrogen, right? What we advocate under organic systems, right? So that's another area where we need to work on. And collaborative links and exchange of expertise, as what we did or we started now, will compare to the previous experiences. And thanks for you for giving us an opportunity, for Sri Lanka, an opportunity to relook at the biology, right, and relook at the behavioral aspects of the tea shuttle borer. And because, I mean, we have been not forgetting, but we have been neglecting that area for some time, because we don't see much new pest scenario. But certainly with your initiation now, certainly we'll get together and look at some more on that. So with that, ladies and gentlemen, thank you for your attention. Hope I gave you a few comments as an insight to the problem. Thank you.
I'm curious about the fact that the beetle attacks such thin branches on the tea plant in comparison to other tree trunks and, wider woody branches. Is this an unusual aspect of the biology of the tea shothole borer in Sri Lanka, and is it specific to, say, tea? Or do you see this in other crops where you grow, say, a shrub or a bush? Does the beetle also attack the thin branches?
Yeah, of course, on tea, that is the susceptible stage, right? That's the susceptible stage. As I said, the biochemical basis in different sizes of the branch has been well understood, right? And of course, the attractants as well as other compounds, right? But to give you some other examples within tea, we know. I mean, we have seen damages on other sizes of the branch or sizes of the stems, but this is the most susceptible stage. But when you go to avocado or rambutan or any other crop, certainly we get additional sizes or additional stem sizes. Yes.
In tea, do you find that the fungus grows down from? Does the fungus grow at all? Does it spread in the tea plant after? From
no.
where the it's just in the galleries.
No, in the gallery itself. Yes.
Okay.
But recently we encountered another symptomological difference. Just similar to some of your experiences here on the stem. Just an oozing out of some liquid. I mean, dark color liquid. That's all, right? I mean, we don't expect anything to come out from the gallery, but just symptomological differences.
Okay, then one other plant. You had a question. You had durian was another host, and it does reproduce on there.
Yes.
And what was the second one?
Rambutan.
Okay.
I mean, just close to lychee. Yes.
Okay, one more quick question, and we move on.
So, do you do a lot of pruning on the tea plant? So, do you see any attack on the pruning wounds by the beetle?
No. But through pruning, I mean, yeah, we don't get any attack. We don't attract beetles, so we don't get any attack to the pruned cut. Yes.
Ruben, last word.
Has anybody tried injecting or introducing saponin into the tree as part of the organic farming, not only depending on the natural occurrence of the saponin in the tree?
You mean injecting insecticide, right?
Just saponin.
Oh, right, okay. No, I mean artificially spraying saponin.
Yeah,
No.
spray or inject systemically, no?
Sampath, do you have any experience? No. But
now, tea is, we consider tea as a health beverage, and we are not supposed to inject anything and to modify the constituent. So we consider it is a natural beverage, so we have to go as it is, right? So saponins or injecting of anything or genetic modification is not possible with tea, right?
Yeah, just one comment. I mean, I'll share some of the experience later. What we, as Sampath said, I mean, we do not do anything on the field, right? I mean, spraying anything, but we do study certain incorporation of such chemicals on artificial diet, right? Where we have experiences, as I said, the same phenomenon what we experience in the field, we reproduce the data similarly on artificial diet. I mean with increased saponins, increased polyphenols, increased levels of pesticides, right? That we have observed.
Okay, we should move on. Thank you.
Thank you.
Good morning, everybody. And I'm so sorry that I had a kind of a very interesting presentation, I guess, and it would have been an overview for the short-haul bore problem in Sri Lanka, and I should have done that before Kirti, but unfortunately the technology doesn't always help us, right, wherever we are. So most of the information that I have in my presentation actually covered by Chiri. So it would have been easier for me had I presented with my presentation. So what I wanted to say is that our tea short-haul bore and tea is a totally different system in a totally different environment and based on the totally different geography. But what I wanted to stress is that given the opportunity, given the right conditions, right environment, and the short-haul bore would establish. So that's the importance that I want to share with you. And the things that I wanted to show would have shown that how fortunate we are to have this culprit in our tea cultivation. But anyway, I had quite a lot of discussions with Richard even long before this meeting was organized. So I will give that point only, how I can share my knowledge and experiences wherever you need or whatever, in whichever aspect that you need. One important thing that I should mention is, now yesterday I think Ruben was asking about whether the short-haul bore that you have here will establish in that area, whether it will establish in that area, that kind of stuff. So we have done quite a lot on development biology, and we establish all the development thresholds. So based on that, and you know, most of the people must be knowing that the degree day accumulations, degree days. So based on the degree days in different locations, we can get a rough idea how many generations would be possible with that climate, given the opportunity. So I have the models to run for short-haul bore and assume that, or rather, the short-haul bore in this area would be the same or maybe not, but we can just give a quick run and see, assuming that is the same species here, how the establishment occur in different areas. So that kind of stuff that I wanted to explain in addition to the scenario in tea in Sri Lanka. So I can help in that way, but in addition to that, the situation with regards to the control of short-haul bore in tea in Sri Lanka it's a real pain. Although we have an IPM program in place, unfortunately we have more emphasis is given for the cultural kind of operations. The chemical control offers a little help for many reasons, as Kirti pointed out, that the Sri Lankan tea is regarded as the cleanest tea in the world as far as the pesticide residues are concerned. So we wanted to maintain that status. So therefore, we are really concerned about using pesticides. And on the other hand, the difficulty in even if we have chemicals, the difficulty in the application. You would not imagine that the planting density is extremely high in Sri Lanka. So for a hectare, we have about 12,500 plants, and that translates into your system here about 5,000 plants per acre. Right? And it's extremely difficult with the dense cover, the application. So therefore, chemical controls offer little help, and I can guarantee that, judging by the entomologists that have been working with this beetle for like 100 years, they hinted that short-tailed borer is not a good candidate for biological controls. So therefore, biological control also offering little or no help. So the cultural practices that we have is in the form of choosing the resistant cultivars, and we have wide array of cultivars that are recommended for different regions. And I had a nice couple of stories to say even how we have to live with this plant resistant business. So we have cultivars for obvious reasons that people go for cultivars with higher yield potential, and also for the cultivars which produce quality tea. So quality and the quantity both matters at the end of the day. Unfortunately, the cultivars that are good yielders and the cultivars that produce quality tea equally susceptible to short-tailed borer. And in the areas where people grow both these clones, and I have the kind of results there. So in certain areas, the plantations, many plantations have more than 80% of their extents under these two clones for that reason. So, and if that area is good for or conducive for the short-tailed borer, so you would imagine the situation would be. Right? So we have that kind of problem with even with plant resistance and things like that. And chemical control is not good. So therefore, we definitely go for this plant resistant business, even though we have this kind of damages in those areas. And the other thing is that the tea plant is, it's pruned regularly every 2 to 3 years, depending on the growth and the elevation. So once we prune that, if there is a damage left on the prune cuts, that will facilitate the further entry of wood rotting organisms, and we have another serious pest, the livewood termites. So it will gain entry and the damage continues, and that will lead to a definite death of the bush. So therefore, one of the things that we recommended is to clean the prune cuts until you get a clean prune cut, and apply kind of wound dressings. Again, that is also not possible for the reason which I was talking about, the extent. So applying wound dressing to a 12,500 hectares is not going to do good, right? So therefore, we are pressed with the recommendations and things like that. So it is very difficult for us to control that pest. So this is the message that I want to put across. This is a very difficult and cumbersome pest to control. So I wish that it would not be the case here, but be prepared for that. And what we have to do before it gets out of our hand, we should do it, and we should act right now. And finally, what I want to say is that the short-tailed borer, whether it's Eualacia or Xylobora or whatever, it's going to be a global problem in time to come. So we have to act upon right now. Thank you very much.
I would like to acknowledge the people that really have done, the work, and basically here transmitting some of the work. And even though the title says just to talk more about the life cycle, I'm just going to go the different areas of what we miss or what we really don't know about Salivoro's glabratus. As you know, Salivorosus glabratus is originated, or the places where it has been collected has been from India, Myanmar, Taiwan, Bangladesh, Japan, and perhaps other countries in Asia, and several hosts are listed, Lydsea, Shorea robusta, Lauraceae, and Fagaceae. However, our studies for Salivorosus glabratus have been really minimal in the area of origin. A very short survey in Taiwan where we placed logs of several of the species that I mentioned before were negative, capturing any Salivorosus glabratus. However, after looking at one of the host plants in a monosmophyllum in a collection, we finally found Salivorosus glabratus. But in the collection, the only time that we found glabratus was when glabratus was associated with termites that had been damaging the trunk, or with longhorn beetles that they were damaging the upper part of the tree. So from that point, even though there were really short associations, we really don't know much about the role of Salivorosus glabratus in the area of origin. Other work that we have been doing relates to avocado cultivar. Both Mayfield tested West Indies, Guatemalan and Mexican races of avocado. And since we have so many avocado cultivars in Florida, around 20-something, what we did was to place them in a no-choice experiment in contact with Salivorosus glabratus. And then we measured the number of holes per stem. And then with Jason Smith's lab, measure the frequency of lauricola. The interesting thing is the beetle bore into all of them, but the interesting part is lauricola didn't appear in three of the cultivars. Again, the trees that were used were small. I think this needs to be done again to verify why lauricola did not appear in some of those cultivars. So there are some indications, but again, some of these studies need to be done again. Other study that we did, and this is in cooperation here with California, was to expose logs of different species of Persea to Salivorosus glabratus in areas where Salivorosus glabratus was found, as well as placing them in the lab in contact with Salivorosus glabratus. Well, I was very happy during the first trials because there was at least one species that Salivorosus glabratus didn't bore into it. Then we asked Marilu Arpaia to send us some more fresh logs, and well, but the possibility of Flocosa not being a host for glabratus was something false that we were following after. So in conclusion, there is a lot that we need to learn about Salivorosus glabratus and the Persea species here in the tropics. This study started, still is, a study in which we were collecting trees, exposing trees that they are common in the Florida hammocks and also in the swampy areas. And the goal for this experiment was to see that there was any repellency. My colleague Lucas Stelinsky also was sampling the same trees and seeing if volatiles from those trees would act as repellents from Salivorosus glabratus. To our surprise, bald cypress, Taxodium distinctus, and Annona glabra, that is a common tree in swamps of the Everglades, we obtained boring of Salivorosus glabratus. However, the lauricola was not positive for two of those. We did not inspect that the galleries was what we call successful galleries or not. But I think the good point here is that if these two were attractive or the beetle bore into that, it might be that some of these trees have some volatiles that can be used even for trapping, and that's what Stelinsky's lab is doing right now, trying to detect which volatiles are present in these ones and if they can enhance the current trapping situation for glabratus. With the behavior, we learned something really interesting. What we learned after observing the number of beetles that were trapped from 7 a.m. until 7 p.m. in the evening, is that the beetle has what we call unimodal flight behavior. And this was the first test. And the second test also shows that during the dusk or close to the dusk areas is when you start collecting this large amount of beetles. And the correlation coefficient was negative regarding solar radiation. So when the solar radiation is becoming less and less, then is when you will trap more Salivorosus glabratus. What's the practical use? The practical use will be then for chemical control if applied at that time, when there are going to be more beetles around, and also for the growers when they are going to do pruning. If a grower is going to do pruning, it's advised that they will do it in the earlier hours in the morning where less Salivorosus glabratus are present. The seasonality, Jim Hanula did seasonality. This seasonality was done in Red Bay and Swan Bay in Florida. And what it shows basically 6 to 7 generations per year. The lower peaks here, I think they are related that the hammocks were exhausted of beetles population, but you still can see some peaks in there. So basically, to me, it will be around 6 to 7 generations per year, again, in those hosts. But I would say that February to March will be perhaps the time where the highest peaks and perhaps when dispersal is occurring at the maximum time from one host to another during the year. So those are the practical implications of that. The life cycle opened a can of worms, really. We tested studied life cycle in avocado, Red Bay, and Swan Bay. And the reason that we started testing this basically is basic knowledge that we needed, but also because in the collections of Red Bay, Swan Bay, and avocado, we always observed that from avocado, very few Salivorosus glabratus were emerging. At the beginning, I thought that was competition with other beetles. Therefore, logs of these species were exposed to 20 females of Salivorosus glabratus. Water was provided, and the logs were split daily to study the development of this insect. Oh, pardon me. This is kind of really interesting, and I have to come closer here. If you see here is the egg deposition, and this is for Swan Bay. Here you have Red Bay, and then you have avocado. You can see that in avocado, like the lower egg deposition, and as well, if you start going to larval development, pupa, and also the adults that they are emerging from avocado, you can see that these are more or less than what emerge per bolt of avocado. So there is something with these two species that logs of avocado lack. So if we go now for the number of successful boring of the beetle into each of the species, we really don't have any differences in there. But when the number of eggs are deposited, you can see that it's similar between Red Bay and avocado, but Swan Bay has approximately 3 times the numbers that you obtain from both avocado and Red Bay. And the percentage of adults that reach adulthood is only 12% in avocado, is 48% in Red Bay, and 90% in Swan Bay. What's going on? There's a difference in all of those cultivars that we don't know what's causing that, but I would like to talk more about that. But in general, the eggs hatch from 7 to 9 days when the temperature is around 96 degrees centigrade, the larva between 6 to 10 days, and the teneral, then the pupa between 4 to 7 days, and then you obtain the teneral adult. And these are the different characteristics. The egg, of course, is ovoid, clear looking. The larva, characteristic of a scolytine larva, C-shaped, legless, and the pupa exarate. 3 larval instars, which is the common denomination for this species. And we also started an experiment that has not concluded right now, but the interesting thing is that around 12 degrees centigrade, we did not obtain an egg hatching or egg development, and pupal development did not occur at 12 degrees centigrade, which calls my attention in there. But the fastest, again, temperatures higher than 36 degrees centigrade, we did not obtain pupal development, and the same went for egg hatching. So there is a difference in here, and I was surprised with the data because when we observed the populations of glabratus in Taiwan was in high mountains, what we call coffee growing area between 18 to 22 degrees centigrade. So I assume that in those places, glabratus was going to show a really good development even at low temperatures, but that doesn't seem to be the same anyway. This experiment has not finished yet. The gallery pattern is very interesting. We have a primary tunnel that is regularly 9 millimeters in length, and then they start branching to secondary tunnels and tertiary tunnels. Most of the eggs are found in the communal area. Sometimes a single egg is deposited, sometimes in groups. So this is found between the secondary and the primary tunnel. In the tertiary tunnels, we did not find the larva, which called my attention what we found in here. And regularly, the gallery length sometimes can go to 20 or more centimeters, and double the time in the width of the gallery length. In other systems, it follows this same ramification more toward one area than to another, but continues showing the primary tunnels, secondary tunnels, and tertiary tunnels. The gallery pattern was studied in Swan Bay and not in avocado. I wonder if the gallery pattern in avocado is going to have the same length that what we found in this host. Fungi in the galleries is a work that is in progress. Harrington and others are found in the Micangia, Lauricola, other species of Rhiphalea, Arce, Subalba, but we really don't know which are the fungi that are found in the galleries at the different stages of development of that gallery. And Jason Smith is working with my student trying to determine which fungi will be present in which gallery. So these are work that is in progress. For me, the advantage to know this will be what's the nutritional value of those fungi. If we're going to find in the galleries the same fungi, I imagine that the ones that we're finding the Salivoro's glabratus Micangia is something that we need to find, but I still think this will be very good for diet development and to see what's the nutritional quality of this. Avocado to me then is still a poor host, and I question why is avocado a poor host? Could be some of the chemical compositions present in avocado that are not allowing the fungi to develop as well as might develop in the other hosts. I don't know if that's the case or not, but I think that there are some practical implications in there. Emergence starts happening around 60 days after the female star completes their development, and proceeds to happen with peaks between 120 and 150 days after invasion of the log, and continues to 240 days after. So I have a very long pattern of emergence that is found. This is what we found in Swan Bay. We haven't measured what's the pattern of emergence in avocado. Large rearing will continue to be based in what is the normal situation. Just collect red bay or Swan Bay infested logs, and we place them in containers in which we put a beetle collecting partition baited with a the manuka bait, and some paper towels that they are wet to maintain a high production in the laboratory. Now, the isolated colony can be produced, but I won't advise to use avocado to maintain a colony because it's a really poor quality host for Salivoro's glabratus. So meridic diets, we have started many, but that is, I call it my fault, because anytime that we start doing that, I pull out the person that is working in the meridic diets, and we never have been able to determine if the diets that we have used, in which we have used red bay or avocado sawdust applied to it, are successful or not. So far, with the latest diets that we are trying, we are finding Salivoro's boring. Still, until last week, no fungal growth in the galleries observed. and other ones we found boring egg deposition in larval present, in which I believe there is some presence of the fungi. So in conclusion, first of all, the surveys need to be started really and continue in the areas of origin to give us a more clear picture of what's the role of Salivorosus glabratus in the area of origin. The nutritional value of fungi in the galleries, I think, need to be determined. The meridic diets we need to develop for further studies. And again, the low production of adults in avocado, that could be the Achilles heel that we might be able to use to avoid a higher epidemic in the avocado industry in Florida. That's it. Thanks.
Just to follow on about your point about the low adult survival in avocado, presumably 12% is pretty optimal, and in real life it'd even be lower than 12%. Do you think Xylembris glabratus populations could sustain itself without, in avocado alone, without these alternative hosts like red bay and swamp bay around? Like, for example, in California, where you don't have those hosts there, do you think it could sort of reach, you have a self-sustaining population just in the avocado?
Well, it is obvious that the infestations that we are having right now in the commercial area are coming from the natural hosts, regularly swamp bay, that is present close to the commercial area. And so that's what the higher populations really are. Now, regarding California, California bay laurel is a host for Salivorosus glabratus. When we expose logs of bay laurel to glabratus, they was born into it. Paul Kendra and Bud Mayfield has pursued those studies and have verified that it does very well on bay laurel. So even though you don't have Persea borbonia or palustris here, you have other susceptible cultivars for the population will build up more. So there is a possibility. So don't discount that.
Jorge,
Yeah.
You mentioned that one thing I didn't understand from your statistic that you put beetles on a log and then 60 to 240 days during that period you would get new beetles emerging. Is that
between 60 to 240 days you get emergence or beetle emergence?
So that is all from, let's say, you put in one mother and then her offspring reproduces on that log again too?
Yeah, yeah.
Oh, okay. All right. No, I thought it was just one mother.
It was there. Yeah.
This is a follow-up question to people who know the botany here in California. I'm wondering about the bay laurel. Is it occurring in the areas around the avocado orchards?
Occurs everywhere.
Anybody?
Sure, yes. It's, especially in the coastal area of Santa Barbara, there is a lot of bay laurel in all the canyons. And it's right next to avocados in many places.
Okay.
One other question?
What is the explanation for the poor performance of the beetle on avocado? Is it related to the poor performance of the fungus, or the poor quality of the feed of the fungus as a feed?
Well, again, when we collected trees that have been affected by laurel wilt through Florida. From those trees, the whole trees were collected, put in cans, and we waited for emergence of Salivorosus glabratus. In all the cases, in some other places in Florida except Homestead, very small population of beetles emerged from avocado. These were from the trees that were infected. Again, at that time, my belief was that there was competition with other scolites. But now, from what we exposed a single bolt, of course, it's a dead bolt, and the tree might have deteriorated. I believe that there is something in avocado that avocado lacks or have that is not helping the growth of the fungus, but I leave that to the pathologists to find if there is something or biochemical, or is the substrate or nitrogen, if the fungus is doing well in nitrogen. I don't know if the amounts of nitrogen in the stem of avocado are less than in the other hosts. That I don't know. I think that's an area for more studies.
Okay. Thanks, Jorge.
Okay.
Our next speaker, we're going to continue the discussion with Raffaella Loricola. Daniel Carrillo will be speaking about ambrosia beetles and the fungus. Daniel.
All right. First, I would like to thank the organizing committee for this wonderful opportunity, and I also want to acknowledge the co-authors of this paper. There's three here in the audience. Alina Campbell, that is doing some molecular analysis to the fungal isolates. Dr. Randy Pletz, that is the head of the pathology laboratory in our station, so he's leading the pathology part, and Dr. Jorge Peña, that is the lead of the head of the entomology lab and the head of all the entomology part of this study. So I guess that by now everybody knows that we're dealing with this invasive species, this ambrosia beetle, Hyliborus glabreus, that vectors phytopathogenic fungus, Raffaella loricola. And when the beetle inoculates the fungus in trees, mostly trees of the family Lauraceae, it causes the laurel wilt disease. So this has served some motivation for us to start studying the ambrosia beetle communities that are associated with avocados. And I don't know if you are familiar with the avocado production in Florida, but most of the commercial avocado production is concentrated in the southern part of the state. There are few avocados throughout the central and northern part of the state grown isolatedly in backyards or this kind of setting. So as the beetle moved south and mostly on swamp base, reproducing on swamp base and red base and native plants, it also affected these avocados, and we tried to gather all the possible information from that, collecting samples and placing them in these emergence chambers and see which beetles we recover from that. The detailed information about this is going to appear in a paper that is coming out in September in the Florida Entomologist, so you could find some detailed information there if you are interested. But in few words, we have identified 17 species, so you see the situation is completely different to what we observed yesterday. Have a great diversity of beetles associated with avocado there, and I'm not going to talk about all of them. I'm going to mention what we have seen with Euwallesia fornicatus for obvious reasons, because you're interested, but our main target is Hyliborus glabreus, and I'm also going to talk about these other species that are the most abundant found associated with avocados. So very briefly with Euwallesia fornicatus, there was a previous record back in 2002, a sample brought to the ornamental entomology lab from our station on a dead branch of Royal Poinciana. And the biologist there, Holly Glenn, thought it was Xylosrandus crassisculus because they have similar size. But she decided to send the samples for ID, and that turned to be the first USA record for this insect. So after that, there was a lot of excitement. They placed a lot of traps in the area. It was a residential area in the Kendall area, and they found some specimens, but not too many, and things kind of cool down. And now in this project where we were collecting wood from avocado, we found a single specimen back in 2009 in a commercial avocado grove. And very recently, like 2 months ago, we found 2 groves that were infested with the beetle. We were lucky that these 2 groves are from one of the growers that collaborates with us a lot. So they helped us they allowed us to put several traps. So now we know that the beetle is widely distributed in those 2 groves, but a common pattern is that we are observing the beetle associated with trees that are already wilted, that died presumably because of other causes, but we are not observing what you guys are having here. Well, that's pretty much the stage of it. We know where the populations are, and we're going to keep an eye on that. But back to our main interest, that is the red bay ambrosia beetle. I think Jorge already touched into this. Basically, we found that Celeboreus glabratus in very low populations, or we didn't recover it in most of the avocados that were affected by laurel wilt throughout Central and North Florida. Finally, in 2011, we found a large infestation very close to the commercial avocado region. It's less than 10 miles to the northern part of the avocado region. We were expecting something to happen. It stayed 1 year with a lot of expectations, and finally, early this year, we found the first avocado that was diagnosed, where the pathogen was recovered from it, but we did not find the beetle. And so far, we have 27 reports. This is in the last 2 months, and we have collected the beetle wood samples from all those trees, and we still do not find the beetle, the primary vector. So we have this wood that is infected with the pathogen. We have this great diversity of beetles, but we don't have the primary vector. And so we started to ask what is the role of all these other beetles in there, and we had one report from Dr. Harrington where he recovered Raffaellia lauricula from Celeborinus saxisani. So we said, let's explore those possibilities. And we selected 9 species of Celeborini. Of course, Celeboreus glabratus, that is our positive control. Celeboreus affinis, Celeboreus volvulus, Celeboreus ferruginus, Celeborinus gracile, Celeborinus saxisani, Silosandrus crassiusculus, Ambridosamus devexulus, and Leconte. And we basically waited to get some good cohorts of each species emerging from wood that is infected with Raffaellia lauricula. And we take part of those beetles to test for presence of the pathogen, to see if the pathogen could be isolated from those beetles, and another part to conduct controlled infestations on healthy avocado and red bay plants. So the part of the recovery of the fungus from the beetles that was carried out by the pathology lab, but in very few words, we just provided the beetles. The beetles were surfaces sterilized and plated in a selective medium, where we could recover the pathogen and count the number of colony-forming units, so we have an idea of the inoculum that is carried by each individual, and then confirm the identity of those isolates with microsatellites and LSU analysis. And this table here summarizes the results of this first part. Here you have the number of beetles that were tested for each species, the number of beetles that were carrying the pathogen, and the probability of a beetle carrying the the pathogen. So to our surprise, seven species out of the 9 that we tested, at least one beetle was carrying the pathogen. And in this, we had a species where very few beetles were carrying the pathogen, like Crassusculus or Saxesani or Affinis, but we had, for instance, the 3, Sallivorus bulbulus, Sallivorus ferruginus, or Saliverinus gracilis, where more than half of the individuals were carrying the pathogen. And if we look at the number of colony-forming units, it was more than 1 order of magnitude higher in the Glabreus compared with the others. But if you look at the range, we found some individuals that were carrying high amounts of the pathogen. So the second part was to conduct control infestations with the separate beetles. Basically release a number of beetles in sleeves and allow the beetles to attack the plants, and then observe if the pathogen, if the disease will reproduce on those plants. When the plants did not show symptoms, we let them for 3 months, and when they showed symptoms, we observed the disease development, and when the plants have wilted completely, we will dissect the plants. And we basically removed the bark of the plants, and we looked for boring attempts on the bark, and how many of those boring attempts passed the bark section and went into the xylem of the plant where the fungus grows. So we looked for internal symptoms, external symptoms, the length of the galleries to see if we could recover beetles or any stages from those galleries. And of course, we tried to recover the pathogen from wood of those trees. And this graph here shows how were the boring attempts and the galleries. You can see that Xyloborus glabreus has similar number of boring attempts and construction of galleries in both hosts. Did well in both of them, but all the other beetles, in general, they attacked both avocado and red bay, but the number of galleries constructed was significantly higher in red bay than in avocado. That has something to do with what we were observing yesterday with these secretions that avocado has. So, and this graph here describes the recovery of the pathogen and the development of symptoms on those plants. So all plants that were infested with Xyloborus glabreus, we recovered the pathogen from all of them, and they all showed symptoms. And in avocado, only we recovered the pathogen from plants that were infested with Xyloborus bulbulus and ferruginus. In Xyloborus bulbulus, we recovered the pathogen from 3 plants, but only 2 were showing symptoms. And the same happened with ferruginus. We recovered the pathogen, but the plants did not develop symptoms. So apparently there is a threshold or a minimum inoculum required to trigger the disease. I think that Dr. Pletz has found similar results inoculating the plants with different concentrations of the pathogen. But in red bay, the situation was completely different. All of the beetles were able to transmit the pathogen and cause the disease. It was variable, but we had the same situation here with ferruginus, in which plant, one plant that where the pathogen was recovered but did not develop symptoms. The same with Saxesani. And with Crassusculus, we had the opposite condition. We had 3 plants that wilted, but we recovered the pathogen only from 1. And we attribute this most physical damage. So remember here that first, Xyloborus crassusculus is one of the biggest beetles, constructed long and many galleries, so we attribute that more to physical damage. So to conclude, well, the biological invasion by Xyloborus glabratus and Rafaela lauricula has resulted in new beetle fungus associations. It's a case of lateral transfer of this pathogen among several ambrosia beetles that are sharing an ecological niche. We know that at least these 6 Xyloborini species, other than the red bay ambrosia beetle, can carry and transmit the pathogen to healthy avocado and red bay trees and cause laurel wilt disease. Of course, this was under controlled conditions, and our data suggests that this phenomenon is more likely to occur in red bay than in avocado. And this opens many questions. Now we need to explore what is the nature of these beetle fungus associations. Are the beetles going to benefit from this association? Is the fungus going to benefit from the association? We need to answer those questions. Will these other ambrosia beetles attack healthy plants? We have some beetles there, like Xylosandrus crassusculus, definitely attacks healthy plants. There are some reports of Xyloborus affinis, Xyloborus ferruginous attacking healthy plants. That's controversial. We have some experiments carrying out. Would they be attracted to stress plants? We are working on an avocado production, a commercial setting, where plants are subject to a lot of stress. We demand from those plants to produce a lot of fruit. Also, what will be the effect of pruning? That is a common practice in Florida. And also, where is Xyloborus glabratus? Are we going to find it in the commercial groves or not? And with that, I finish. I would like to acknowledge the Florida Avocado Committee, the Florida Department of Agriculture and Consumer Services, and providing funding for this research, Mike Thomas for the beetle ideas, and everybody in Peñas and Pletz's lab for working in this project. And thank you very much.
You showed quite a few images of plants in containers. Are you seeing it in plants and containers at nurseries in the Florida area?
Can you repeat that?
You had a lot of images where you showed plants in containers, like from nurseries. Are you seeing it, the beetles?
Oh, no. No. Not in the nurseries. And we have not seen the beetle in the avocado growing region at all. No? So far it's been up to that large infestation near the avocado growing region, about 10 miles north, but we have not detected the beetle in that area. And that limits a lot of our research because we cannot work with big plants. All the research is limited to these small plants.
I just wanted to mention that experimentally and in the field, there's a profound correlation between plant size and whether or not this disease will develop and whether the beetle is attracted to trees. Bigger trees, more attacks, more disease. Small trees, not so much.
Okay, thank you, Daniel. You're welcome. Our next speaker is V. Mendel, who will talk to us about avocado wilt in Israel.
Thank you, Bob. First of all, I would like to thank many people around here in the States and in Sri Lanka and Israel for sharing with us their knowledge, their experience, their thought about the situation with the shoot-all borer. I'm going to concentrate in this presentation on some aspect of the biology and behavior of the beetle. And I was lucky to have two other presentations where I will tell you something about the chemical control of the beetle and the situation in Israel. Well, this is a volcanic center, and from here, Stanley and I start to work on the beetle and the fungus. So first of all, I would like to show you the list of hosts that managed to be attacked by the beetle. And I'm separating between what we call a suitable host, 4 species, and I listed them according to their susceptibility to the beetle and the fungus. We don't separate between them for the moment. And a short list of other trees which are intercepted by the beetle, but we don't see any development. And about the recovery of the fungus, Stanley will tell you a bit later. But what I can say that we can easily distinguish between different type of non-host. Some of them are highly attractive for the beetle, others are not really attractive, and I will suppose that they were intercepted by the beetles simply because they are growing in adjacent, very close to infested trees. Well, this is the elder box in one of our net house where we do some of our experiments. And if you look at this penetration spot, you see the typical development of the fungus only on the xylem, and the cortex is relatively healthy. Actually, it's healthy. You don't see any sign on the cortex. This is what happened later on. The tree responds, and you see the pitch out of the gum, which is typical to elder box. Here you see the xylem of infested and healthy elder box, and you easily can see the spread of the fungus inside, whereas the cortex remains rather clean. Elder box is extremely sensitive, and whenever it's infested by the beetle, the tree dies very soon. Here you see one of the specimens in Tel Aviv. Actually, all elder box in Tel Aviv were killed by the beetle. There are not very many of them, but the beetle managed to find each one of them. See again another view of this elder box. You see that the low part of the tree is still alive, and somehow it seems they first of all they attack the upper part of the tree, and then they move to the low part of the tree. Another important host is the custard bean plants. Here you see one of the small trees which are infested. It's easily to distinguish between healthy and infested trees by looking at the upper part of the canopy. You see dry branches. This is a very good symptom. And then look down and you see those penetration spots, usually in the thick part of the stem. You don't see any secretion. Nothing comes out like a gum or anything. It seems that the tree doesn't respond to the penetration of the beetles. And then again, you cut the tree, you look at the xylem, and then you see the same picture, infested xylem, and compare with a healthy one. Here easily you can see the penetration spots of the beetle. Another thing which is very interesting is this black stuff that covers the walls of the gallery. You can see that in every infested host. We don't know exactly what it is, but it's very quite typical. Here you see again a closer look of the galleries in custard bean, and again you see this black stuff that covers the wall of the gallery. A closer look, you see the eggs, which are very many, by the way, in custard bean plants. Again, this is another gallery in the custard bean, and you see this. The gallery is long, and you see bunches of eggs, which are very typical, and the larvae. You can see the fungus, and again, there's black stuff that covered the the walls of the galleries. Again, another picture of the custard being infested by the beetle. Another host is English oak. There are several species of English oaks. This particular species seems to be, at least in Israel, extremely sensitive. And on many occasions, we found that these oaks are dying because of the attack. Closer look, you see the cankers all over the branches. And closer look, you see that the beetle also colonized the twigs, or not exactly twigs, the small diameter branches and medium diameter branches. And in this case, the typical symptom is wetting, some kind of wetting on the bark. And again, a closer look on the infested branch, and when you look inside, you see the spread of the fungus along the xylem. And again, typical cankers that are, you can see in other cases, like in the black walnut, for example, you may see a similar situation of small cankers on the xylem. Again, this is one of the non-suitable hosts, Brachychiton species. Again, you see again the pitch out of a gum here. You see that all over the tree. The trees are infested whenever there are suitable hosts around. All these trees grow in adjacent to other susceptible hosts, usually elder box. In some cases, in some cases, the tree dies. We don't know why. I'm not very sure now that this death is directly related to the infestation by the fungus, because we don't see any beetle coming in or develop in Brachychiton. Okay, like a tree like this, a street tree, which deteriorate very quickly after the attack by the beetle, but you don't see any beetle inside the tree. Oh, sorry. Okay. Now we turn to the avocado, and you see the very clear symptoms of persitol exudation on the bark, which we already saw yesterday. And again, in heavily infested plantation, this is what you expect to see. A lot of cankers, which black at the center, the white sugary exudation around this black spot. Very typical, very unpleasant. And again, usually you see the first infestations inside the canopy. This is a bit older one. This is a bit younger infestation. And first of all, you see this sugar eruption, as you call it, and later on it's vanished, and you see this typical canker. Sometimes the cankers vanish or start to decay, like you see here. In most of the cases, the canker grows as long as the beetle are still inside. So this is a big canker, and it's continued to spread until a certain point. Again, and the beetles tend to recolonize a section of a branch or a stem where it was already occupied by the beetles. It's certainly not colonization by the brood, because the brood didn't emerge yet, but other beetles join those infested sections. Here, for example, you can see one of our observations when we compare the situation after 20 days. So we look at the situation of certain branches that we marked in particular date, and then we came back after 20 days and we count the number of penetration spots, and you can see a very clear correlation between level of infestation and additional penetration spot later on. However, this is not always the picture, because if you look at this situation, and you still see some correlation between the level of infestation and additional infestation spot, but the correlation is not very good. And there are some cases that you don't see any correlation at all, like in this particular Haas young plantation. And again, we measure the relationship between what happened before and what is the situation later on, and we don't see any correlation. So there are different situations. We don't really understand what makes the difference between the different situations, but we still study that. And later on, of course, you see something like this, and then you see different kind of response of the tree. In some cases, you can hardly see any exudation of the persitol on the trunk. In extreme cases, other extreme cases, you see a lot of them. It seems to me that this is somehow related to the condition of the tree. This is a poor tree, and this is a better tree with more vitality, and that's why the tree responds like that. Again, so we cut the tree just to show you what is inside, and this is what you can see. This is the upper part, the outer part of the bark, and you see inside, you see the spread on the fungus, and again, you see that the cortex is actually clean. And then the tree is alive, by the way. Well, inside the xylem, that's what you would see, what you expect, the beetles in their galleries, and you can count the eggs if you wish, if you're lucky enough to cut it in the right section. And sometimes you can see the old stages inside one a piece of branch. You can see here, you can see the adults and the callow adults and the pupa and even eggs. Well, this is the typical position of a larva. Again, you see this black stuff that covered the wall of the gallery. This is I would say, the late stage of the third instar larva. And here, what you can see is a cut in a branch again, and you can see the galleries. This is just before the emergence of the adults. It's not a very good photo, but again, you can see a lot of adults around here. You can see the galleries, and again, you can see this black stuff that covers the wall of the galleries. This is emergence, and only in these cases you may see some frost coming out from the branch or from the stem. Again, we cut the branch, and then you can see here inside the typical gallery and the typical infestation of the xylem. And again, you see this is a branch that breaks probably because of the weakness of this point due to the galleries inside. Of course, we can easily rear the larvae on the petri dishes on the PDA that we received from Stanley, and you can see very vividly the larvae feeding on the fungus, and they like it very much, by the way. And later on, they get into the cortex and constructed the gallery inside the petri dish, and you can see the galleries inside a petri dish, and you can see the male and the female inside those galleries, and even the eggs. And then they managed to, not very many of them, but they managed to complete this cycle in a petri dish. Here you see a callow adult. Actually, we found out that they don't tend to lay too many eggs in those petri dishes, so when we add sawdust of avocado, we had managed to increase the number of eggs that were laid in the system. It's still not very satisfactory because the production is not very good, but again, if you add the wood material into the system, you improve it. So this system also allows us to do some measures and to follow the development of the beetle inside those petri dishes. So here we compare the development of beetles that were taken for two different sources, beetles that grew on the petri dish, on the Fusaria on the petri dishes, and beetles that we extracted from avocado branches. And what you can see here is that you don't see much differences between the development stages that actually their parents were adults of those two sources. However, if you look at this, the time from introduction to the position was much longer if the adult beetle were collected from the petri dishes. And that's, of course, influenced the entire development from introduction until the inclusion, until they became adults. By the way, we did, as a control, inoculation or colonization of disconnected branches of avocado that we keep in water. And you see that the development of those beetles, of course, we couldn't measure the development of the immature stages, but you can see that the development of the beetle in those branches and in the petri dishes was more or less the same. Of course, we tried different methods to monitor the beetles. Eventually, we found that it is a waste of time for the moment because it's very easy to spot or to detect those infestation spots because of the exudation of the percival. But anyway, we tried manuka oils, of course, and you can see here, this is a photo of a plate with the beetles on those sticky panels that we hang in the avocado plantations. And we compared the, of course, sticky trap with the bait, without the bait, and different colors. Here you see the results, the differences between red plates and white plates. And eventually, of course, the manuka oil affect the attraction, of course, and we get more beetle with the bait. And it seems that the red plate are somehow better than the white ones, but since it's difficult to count the beetles on the red plate, we prefer to work with the white ones. But again, it's not very promising methods. And I would like to finish my presentation with some information about the seasonal history of the beetle. And what we did here is we studied the state structure of the beetles inside the branches of avocado and custard bean plants in different times, last year and this year. And you can see quite clear that in most of the time you see adults in the galleries, but that gives us some information about the dynamic of the beetles and the number of generation. We compared that with information we managed to generate by suspension of those sticky plates, and we know by now that we have about 3 main peak flights during the year, which more or less correspond to the state structure of the beetle. And that's all. Thank you.
So, hi, Zvi. Thank you very much for this presentation. I have 2 questions for you. One of them is on the brachyhedron tree on the street. You mentioned that that's not a suitable host for the beetle because you didn't see the beetle, but tree is still dying. So, have you isolated fungus out of that?
No, not at all. We were overwhelmed by the different tasks we needed to do, so we couldn't pay much attention to that.
You couldn't look at it. Okay. The second one is, so in Israel, you have this problem in your commercial avocado growth. So can you tell us a little bit, what is the economic impact of this past disease on commercial avocados?
Well, I will refer to that tomorrow, but we don't have good figures by now, but the situation is bad.
Zvi, you showed really good correlation between previous attack and current attack. And in some of those relationships, there was no relationship. But I noticed that in at least a couple of those graphs, there were different cultivars. Was there a cultivar relationship where on Haas, you didn't have a relationship? In Edinger or another cultivar, there was that relationship? Is there a genetic component there?
Yes, I will refer to that tomorrow as well because of the limit of time, I didn't address the issue of difference between different susceptibility between different avocado cultivars. But some of the cultivars seems to be not very susceptible, although eventually after repeated attack, they will become infested as well. Haas, for example, is extremely susceptible. Ettinger is almost not susceptible.
Two quick questions, Tzvi. Excellent talk. I have one question for you and one question for the mycologist in the audience.
Okay,
right here,
Jerry, okay, sorry.
The question for you is, have you tried ethanol lures rather than manuka oil? Because
yes.
we know that manuka oil is good for the fairly specific red bay beetle, but I'm not sure if...
That's the first thing we tried, actually. And well, I have to mention this was an advice that we received from Bob to use ethanol, and we also added alpha pinene as well, with no luck.
Interesting. And another question, based on those beautiful images you showed of the galleries, we all know that the galleries are black. We also all know that the fungi in media are kind of whitish, pinkish. Does any of the entomologists, mycologists in the audience know what is the black stuff that's in all those galleries?
Well, we can guess, but we don't know.
I would assume that it's probably not the fungus. But
No, it's not the fungus. I believe it's not the fungus, but again, I don't know.
So I would imagine it's a host reaction.
Pretty common, but it's not something that we look at it over and over. And it looks, if you take it and scrape it, put it under the microscope, it's wood fibers, and they're black. So I'm assuming it's a host response. We see it in conifers too.
Thanks again, Zvi.
Okay, thank you.
So I'm supposed to, as discussant, kind of get some of the discussion going here. We need to know three basic things, and these are the three things that I guess as a discussant I'm supposed to stimulate this discussion here. Where is it? What is it doing? And how do we manage it? I think understanding the biology of the insect and the pathogen, if we can help answer those three questions, we would go a long way. So where is it? What do we need to do to survey for it better? Ethanol might not work, manuka oil might not work, but what can we find that are attractants that could help us survey for the insect to figure out where it is? That's one topic. I think, what is it doing? What are the impacts? A lot of work could be done there. What is the host range? What are the interactions with other insects, other bark beetles, ambrosia beetles that we heard from Daniel today? And then how do we manage it? That's the key question. Are there chemical controls, biological controls, host resistance, that sort of thing? So to me, those are the three things that if we could find out some of the biology of the beetle to help us answer those questions. So 15 minutes, then lunch, so open it to discussion. Andy. And I'm not going to answer these questions. I'm just up here.
I don't have a question for you.
Good.
I have maybe a little teeny piece of maybe a good news regarding your first point, which is we need to figure out what these things are attracted to, and maybe we'll be able to use those chemicals, volatiles. We have worked with tested whether the fungi themselves are attractive to the beetles. This was the case of several species in the eastern United States, and they are kind of attractive, even long-range attractive. It's not as attractive as the tree itself, specifically attack trees, but there is something that the fungus is producing that's attractive to the beetle. And so now we're in a stage of figuring out what it is, and it turns out that these are all, and I mentioned that to several of us here, these are all short-chain alcohols, very volatile alcohols that the fungus produces when it feeds, and when the tree maybe is also producing when it's being attacked by the fungus. Not quite clear where it's coming from, but these are alcohols, and so analogs of ethanol, really. So these things should theoretically be easy to manufacture, relatively cheap, and we just need to figure out, the concentrations, the identities of these chemicals, and there may be a good avenue for further research looking at the products of the fungus and how we can use those to attract the beetles.
Have you looked at, has anybody looked at normalized vegetation index or reflectance of different hosts as far as infested versus non-infested plants to see if there's any satellite or aerial photo type of reconnaissance you can do on the spread of the pests?
Not for this particular one that I know of, but it's been done for other bark beetles, but maybe Randy looks like he has an answer.
Yes.
Okay.
Well, two things. Just I wasn't going to even bring this up, but we're working with an ag engineer to remotely sense laurel wilt, and the data so far look promising. I'm not going to say that we're going to be able to distinguish it from everything out there, but so far it looks pretty good. But my other interest, though, in this thing, and it doesn't, I don't think it addresses your 3 points, but the ability to predict these problems. And going back to the T-shot hole bore, is the situation in India the same, where in tea at least, that this thing is not associated with any kind of disease damage? And what about these other hosts? That, with this Eulacea fornicatus thing, would we be able to predict what's going to happen here in Israel and California based on what's happening on the Indian subcontinent? Just if anybody's got perspectives.
Shall I talk? Wow. You respond. Okay.
I'm from India. In India, we have Persia bombycina plant, which is economically important for us because we are getting these plants is for our silkworm. It's and we are getting golden silk from that after feeding from this plantation. So the beetle I found first time in 2011. It's on the basis of the branches, and it was dieback is there. But I could not find any fungus associated with this beetle. So
the article you have shows disease symptoms in those.
no, there is no disease symptoms. Only borers is symptom only boring. And I have the samples, already I have sent the samples. And in some other plants, I found some sugar exudates, but I don't know it is related to that borer or not. Because I did not collect the specimen from the sugar exudates and other.
All right.
Is this on?
This is regarding what Jiri said. Well, Lucas is working with volatiles from Rafaelea. What he has found is that the attraction is similar to Manuka oil. So I think that joining perhaps whatever people are finding, essential oils plus the volatiles from the fungi to see if there is a synergistic effect with the two of them, because so far what we have right now, what is captures the beetle, in this case Salivurus, when they are really high densities, but at low densities, basically is not that powerful. So what we need is a really powerful attractant.
Can I suggest another model of, let's say, host colonization? It seems to me, and let's say a rather simple model is, if these things just fly around randomly, they land on a tree, and they try it out, see if they can get in there. And if it is a suitable one, like, for instance, the box elder, they go in, and the females reproduce. And when her brood emerges, it seems to me that as a beetle, it would be a good idea just to stay on that very same host. Why fly away and run the risk of not finding anything, and simply recolonizing that same host? And in that way, you can get really high densities on suitable hosts without, let's say, the need of any kind of attractant.
I think that's the case, especially if the host condition is still suitable for future generations. They'll probably keep hitting that. I think you've seen that in other ambrosia beetles, right, Jerry?
Going back to attractants, I mean, what I'm getting here from this meeting and from seeing yesterday the damage, it seems that the box elder, both in Israel and in the United States, is one of the clear favorite hosts. And so maybe, I don't know if somebody have looked at that already, this might be a good tree species to look for an attractant that can be applied to both Israel and the United States. And I don't know if somebody else here in the audience already have looked at that, but that might be a possibility. And correct me if I'm wrong, if it's not that particular tree that is a common species across the different countries.
As well as castor bean, I was thinking that seems to be a very good host.
Yep,
So
exactly.
there's some traction there.
Well, I thought about the castor bean till I ran into Rajesh, and in northern India, where he is at, castor bean do not get attacked by this ambrosia beetle, and this is where it really doesn't fit. Here he has an avocado relative, and it doesn't have any fungal effect, and then at the same time, where they experience that in southern India or in Sri Lanka, castor bean is a very good host for it.
Actually, we have all tea gardens in Assam region. So I surveyed the all tea growers and tea gardens there, but I could not find any infestation of the tea soot-hole borer there. Only in our plantation I found the tea soot-hole borer. And I surveyed that castor plantation also, because we have another silkworm, the Samia ricini, which feeds on the castor plants. So I did not find any specimen from castor.
So did we find fornicators? Did we identify fornicators?
Yeah, it was identified as fornicators by Indian Agriculture Research Institute, the taxonomist. He is a coleoptera taxonomist. But after sequencing, came to know it's to another genus.
Right.
Yeah.
So we might not be dealing with Wallacea fornicatus in your case.
Yes.
It might be some other.
Yeah, it's some another. Yeah. And there is I found some other species also, but I have not taken that photographs and specimens. But definitely there are some other species also available. Because I found some in like this Litzia monopetala. It's another host plant, belong to the Lauraceae family. I found some symptoms. After seeing here all the symptoms, then I came to know there will be definitely some other species are available.
Diana had a question.
Thank you. Something to think about, I think, when trying to come up with a place to look for attractants is to go back to the insect's basic biology. For most of these that are sib-mating, that's believed to have evolved to deal with the fact that your hosts are rare. And so if you're going into dead trees or dying trees, in most situations, those are going to be widely scattered. And so if you can mate with your brothers or even your mother, then you utilize a rare resource. And then if that resource stays suitable past one generation, you're going to keep coming back and using that. And so what we see with a lot of ambrosia beetles is that they will return and reuse a host until it's used up, and then they disperse. And so I think if you look at the the volatiles that are released by dying trees, trees that are perhaps being reutilized over time, like Yuri was saying with some of these fungi, that might be your best place to start, to look. This sib-mating thing came about for a reason, and it's probably because it allows you to use something that's relatively rare. If you have to locate a mate and you're dispersing all the time, you're lowering that potential. So looking back to the basic biology of these insects, I think, is critical.
Okay, one last question. Comment.
I just wanted to put into perspective the sort of difference between essentially three different forms here that we're all discussing. So in relative terms, genetically, our California and Israel material match each other. The California and Israel material are different from the Florida material, but they are, in relative terms at least, closely related. In relative terms, the material that we've received from Persea bombacina, from Som in India, is actually quite a bit different genetically. So we're almost certainly dealing with at least 2 species in that, and possibly 3 would be my guess there.
Thanks again to all the speakers. And Mary Lou, you have any words about lunch? Thank you.